Hello friends, welcome to the Biological Life Sciences channel. Today we are going to see properties of DNA. As we all know, DNA, that is deoxyribonucleic acid, is a heteropolymer of nucleotides, that is A, T, G, C, adenine, thymine, guanine and cytidine. So today we are going to see the physical properties. The first physical property is absorption of light. The second physical property is buoyant density. The third physical property is denaturation and renaturation kinetics. Then fourth is solubility, fifth is size, sixth is purity of DNA, and then melting temperature denoted by Tm. So we are going to see all these properties one by one. So let's begin with the absorption of light. So first of all, absorption of light, you know that DNA absorbs the light in UV spectrum of electromagnetic waves. So UV range is from 200 nanometer to 400 nanometer. So in this range DNA is able to absorb the light. Okay, so and the absorption maxima is at 260 nanometer. What is meant by absorption maxima? It is the point at which the molecule, the biomolecule, absorbs the maximum radiation. So for DNA, the absorption maxima is at 260 nanometer. So the DNA will absorb most light at 260 nanometer. Now for the free bases means the bases which are separate, means nucleotides are separate. For the free bases in a solution, the absorption maxima at 260 nanometer is 1.60 units. For single-stranded DNA means the only one strand of DNA is there. For them, the absorption maxima is 1.37 unit. And for double-stranded DNA, it is 1.00 units. So this helps in determining the absorption maxima of DNA. Okay, so let's see the next property now. The next property of DNA is buoyant density. It is also called as density of DNA. It is determined by isopycnic centrifugation. Or this is also called as equilibrium gradient centrifugation. Okay, so in this case a salt solution is used, that is cesium chloride, CaCl. This solution is used for centrifugation. The DNA is mixed in this solution and it is spun at a high speed. So it is the DNA in the solution will settle at a particular band according to its density. So the buoyant density of DNA is 1.73 grams per centimeter cube, which is equivalent to 6 molar of cesium chloride solution. So when we take 6 molar of solution in density gradient centrifugation, the density comes out to be 1.73 grams per centimeter cube. So this helps in knowing that how the DNA will be isolated from different biological samples. So we can frame the solutions from which with the help of which we can isolate the DNA. So this is an important physical property that helps in isolation of DNA. 10% increase in GA concentration, that is guanine and adenine concentration, the molecular weight of the total DNA increases. So what will happen in the isopycnic centrifugation? The DNA will settle at the much bottom, at the lower side. So for every 10% increase in GA concentration, the buoyant density increases by 0.12%. So this technique is so sharp that it can even determine the difference or separate the difference of isotopes also. For example, carbon 12 isotope and carbon 14 isotope. So such a small difference in the weight of the two elements also can be separated using such kind of centrifugation. Okay, so let's go to the next property of DNA, that is denaturation, renaturation kinetics. So here we determine whether the DNA is complex or not, whether there is a presence of repetitive sequence in the DNA, and what is the GC content in the DNA. So there are different steps in determining the denaturation and renaturation kinetics. First of all, the DNA is heated. So what will happen? All the double-stranded DNA will convert to single-stranded DNA, single-stranded form. Okay, so it will start denaturing. Once the 100% DNA is denatured, what will happen? It will allow to be cooled, that too slowly. Why? Because when the solution is allowed to cool slowly, the single-stranded DNA present in the solution starts to renaturate and form the double-stranded DNA. And this will happen slowly because we are allowing solution to cool slowly. So when the solution is cooled slowly, the readings are taken for the renaturation of the DNA. So that readings are taken as concentration versus time graph. We can see here in this, here it will be percentage of single stranded DNA, and here it will be log 10 value of concentration multiplied by time. So the concentration multiplied by time value will be concentration of DNA in grams per mole multiplied by renaturation time taken multiplied by buffer factor. So it is the multiplication of concentration, that is initial concentration multiplied by time. The buffer factor is added here because the renaturation takes place in an aqueous solution, and that aqueous solution is a buffer. So that buffer strength is also multiplied here to calculate the COT value. So this is also called as COT analysis. So for example, at this concentration or this percentage 100%, the DNA will be 100% denatured. So as the slowly time is passing, the DNA solution starts to renature and it will come here like this. So whatever the curve forms here, it shows the renaturation kinetics of the DNA, and this curve is also called as COT curve. But actually here it is concentration, that is initial concentration multiplied by time of renaturation. So at this position what will happen? There will be lots of repetitive sequences. Why? Because repetitive sequences are easier, simple sequences that will renaturate faster. In the middle there will be small amount of repetitive sequences, and here there will be unique sequences that takes time in renaturation. So this renaturation kinetics helps us to determine whether the DNA is complex one, it is having unique sequence, or what is the GC content. If the GC content in the DNA is more, what will happen? It will take a longer time to renaturate. So what this COT value tells us, COT value, if the COT value is higher, the DNA is more complex. More complex means it is having large number of conserved sequences or unique sequences. If the COT value is lower, then it is less complex. So the COT analysis also helps in determining the complexity of the DNA, what is the evolutionary relationship of the DNA, how the DNA is evolved. We can compare the DNA of a same species which was earlier at some time in history and the DNA of the solution or DNA of the organism present now. So using the COT curve analysis, we can say how the DNA is evolving of a particular species. Now let's see the next property of DNA. That is solubility. So as we know that DNA is soluble in water, since inside the cell there is a presence of water, so naturally DNA is soluble in water. But compared to DNA, RNA is more soluble in water, as at the second position in the ribose sugar, there is a presence of OH group, hydroxyl group in case of RNA, and the same hydroxyl group in case of DNA is absent. So RNA is more soluble than DNA. Okay, so DNA is also miscible in water, but there is a certain limitation to it than RNA. And DNA can also be easily precipitated out from that solution using alcohols such as ethanol, isopropyl alcohol, which therefore it is generally used in isolation of DNA. So, and due to the presence of phosphate group, the DNA is also having negative charge. So, more the amount of DNA or larger the DNA, more will be the negative charge, as the phosphate group will increase in that DNA, therefore the charge will also increase. Okay, so let's see the next physical property, that is size of the DNA. Next physical property is size. Okay, so DNA varies based on size. Okay, so how DNA varies? It varies from organism to organism. For example, in E. coli, the amount of DNA will be very small, or the size of the DNA molecule or genome will be small as compared to human, because human being is a eukaryotic complex organism, whereas E. coli is a prokaryotic small bacteria. Okay, so the size of the DNA depends on the genome or the complexity of the organism. Okay, so now this DNA can range to millions of base pairs, and it can be easily separated using agarose gel electrophoresis. Agarose, as we know, is a polymer. It will form a mesh-like structure. So, depending on the concentration of the agarose you take in the gelling, the pore size will be depending on that, and based on that, the different sizes of DNA can be purified. The second way you can purify or you can separate the DNA based on size is polyacrylamide gel. Here also, this is a polymer which is made up from acrylamide and bisacrylamide. There is a cross linking of bisacrylamide. So based on the porous nature or the pore size we want, based on that we can vary the concentration of this, and based on this you can separate. So using this, the size from one base pair of DNA can be separated up till millions of base pairs. Means you can adjust the concentration of high quality agarose to separate one base pair of DNA also, and it can go on till millions of base pairs also. Okay, so this was all about size, physical property of DNA about size. So let's see the next property of DNA. That is purity of DNA. Okay, so in molecular biology experiments, you know that there are lots of techniques to isolate the DNA. Okay, so what happens? No technique is perfect. Sometimes what happens? The purity of DNA is not there. Purity of DNA means what? Its absorption at 260 nanometer. Whatever the DNA solution we have purified, when we take its absorption at 260 nanometer, under UV visible spectrometer and its value at 280 nanometer, this value should come out to be 1.8 approximately. If this value comes out very close to 1.8, then DNA is said to be pure. And if this value fluctuates, then there is a contamination of RNA, protein, or other organic material. We can say that here, if the value of 260 nanometer is the absorption maxima, whatever the value comes out, if it is higher, then there is a proteinic contamination, and if it is lower, then there is a RNA contamination. So based on that, you can check the purity of the DNA, how pure your sample is. Okay. So let's see the next property, that is melting temperature. Okay, so melting temperature is also one of the parameter by which you can determine the complexity of the DNA, or what are the constituents of the DNA, means what is the GC content in that, all the things we can determine by melting temperature. So what is melting temperature? It is the temperature at which half of the total DNA is melted or is in single stranded DNA or randomly coiled form. So it is determined by Tm, and this tells us what is the GC content of the DNA. As the GC content of the DNA is more, it will form triple bond. We know that A forms 2 hydrogen bonds with thymine and guanine forms 3 hydrogen bond with C. So if the GC content is more in the DNA, it will take a higher time to renature, or it will take a larger time to break. So based on that, the melting temperature will vary. This is denoted as Tm. So the more the melting temperature, the more will be the GC content in that, and less the melting temperature, the less will be GC content in that. Okay. I hope you have understood the physical properties of the DNA. In the next video, we are going to see the chemical properties of the DNA. Till then, bye. Thank you. If you like the video, please share, comment and subscribe, and press the bell icon for further videos. Thank you so much.